between NCBS and CFTRA and Kalasurji. This probably is the first time, ma'am, that Science Public Series lectures are being at least organized here in this space, and thanks to all of you, and then we are greatly honored, right, to host this session. Just maybe 30 seconds, we don't want to be very formal about it, right, but then just 30 seconds for those of you who are coming for the first time to this space. Kalasurji has been an organization which is active in the cultural space for about 30 years now in Mysore. We do regular plays, mostly in Kannada, but we also do it in English, at least one play per year. We have been active in children's storytelling sessions. Every Saturday, come here at 4:30. You have storytelling sessions which have been running for 10 years now. We do literary programs, monthly literary programs, and we do at least 4 or 5 plays right in a year. And incidentally, connecting to science, 3 institutions from Mysore, right, Kalasurji and Parivartana and Raghu, have been coming together to host a 3-day science play festival. Right? Perhaps, definitely for the first time in Karnataka, and perhaps in India, which is a 3-day science play festival. And we have had 2-year running, and we are into 3rd year right now. So with all of this, I think we are well placed to hear from the scientists here. I am again honored to welcome Dr. Raghu Padinjat, right, from Kerala and Bangalore. Welcome to this place. And Dr. Varadarajan Sundaramurthy. Yeah, there he is. Right. So these 2 gentlemen are going to speak to us. And without further ado, I would request Maheen and Siddharth, who also have joined us from NCBS. I would request Maheen to introduce the speakers today. Thank you so much.
Good afternoon, everyone, and thank you so much for coming on this very warm and sultry day in Mysore. This is not working anymore, is it? No. But thank you everyone for coming. We are so happy to see such a turnout. It's so fulfilling to be to do this in Mysore. Mr. Koladu Sharma from the CFTRI is an old friend of ours, and it just seemed natural that we bring our Science Cafe to Mysore in a collaboration. Just to correct Mr. Shashidharan, it's not just the NCBS. This is the Bangalore Life Sciences Cluster, which comprises 3 research bodies, which is the National Center for Biological Sciences, INSTEM, which is the Center for Stem Cell Science and Regenerative Medicine, and CCAM, the Center for Cellular and Molecular Platforms. And these 3 bodies form what is known as the Bangalore Life Sciences Cluster. The Bangalore Life Sciences Cluster is what we represent, with Science Cafe, and we bring to you scientists, innovators, entrepreneurs, and people like that who have stories to tell about the science that they do and the science that interests them. So our first speaker today is Professor Raghu Padinjat. Raghu is well known for being a big part of the ADBS program, which is the Accelerated Discovery of Brain Disorders, and works very closely with NIMHANS. And it's one of the foremost projects that is being incubated at NCBS. So he's going to be our first speaker, and our second speaker is Dr. Varadarajan Sundaramurthy, who's going to be talking to us about infectious diseases. I can assure you both of them are seasoned speakers and we'll have a really good time. Just before I sign off very quickly, I want to tell you about the format of Science Cafe. This may seem to you like a science talk. It is not just a science talk. The point of it being Science Cafe is that it's an informal exchange of ideas and concepts, which means you may be sitting down as an audience, but you're not just an audience. You're here to have a conversation with us. So please engage with us in a dialogue. Please feel free to interrupt the speakers whenever you like. Ask any question you want about the work that they're doing or the science that they're talking about right now, or even about them as scientists. It's an open floor. Please treat this like your living room. In many ways, this is the living room. And once again, thank you so much for coming to the Bangalore Life Sciences Cluster Science Cafe. Thanks.
So there's been a power outage. It will come in 5 minutes, I've been told. So if you want to start.
So since there's a power outage, this will be very informal. Let me perhaps begin by saying a few words about myself. It doesn't require slides or any other such assistance. So one of the things I've been told by younger people that I come across is that they're often very unclear about what a scientist is or who that person might be and how did they get to being a scientist. This is a very important question, and although my trajectory to my present position is not very typical, I thought it would be interesting to say a few words about it. So, as was mentioned a few minutes ago, I came to Bangalore when I was 3 years old. And I did all the things that children do, which is to go to school and college and so on. And then when I got to 12th standard, like most kids, I didn't really know what I wanted to do. So I was good at my work, but I didn't have a clear vision of what it is that I wanted to do. So I did the usual thing, which I think these days is called CET, had some other name in the distant past. And then I ended up going to St. John's Medical College in Bangalore to ostensibly train as a doctor. And what was remarkable was that 6 months after I started medical school, I met a teacher who explained to me for the first time in, let's say, 18 years, what the difference between knowledge is and the acquisition of knowledge is. That is, what is the difference between knowing the facts about something and the process by which those facts are discovered. So once I heard this, it seemed to me that this was an interesting enough thing to do. And so, without too much of thought, I decided that research seems like an interesting thing to do. And so I resolved that I would go away after I finished my medical school and do research. And that's how I ended up doing a PhD, which was at NCDS. I'm from the first batch of PhD students of NCDS, which has now been around for 26 years or so. So that's how I got to where I am. Since we still don't have slides, I think maybe I should just start without slides. Is that okay?
Excuse me. We have a seat here, yes,
Wait, we'll continue till the dog comes. This 5 minute thing we don't know.
Okay. So, anyway, I think I can start my introduction to this presentation without slides. I'm sure every one of you in this room has been ill at some point or the other. Now that illness may have lasted for a day, it may have lasted for a few days, and perhaps it may have even lasted a month or so. And unfortunately, some of us are left with lifelong conditions such as diabetes, where we have to manage the illness in a manner that we can lead as productive and as healthy lives as possible without the opportunity for a permanent cure. So when you go to a doctor, the doctor looks at you, he tries to make a diagnosis, and he will then offer you a treatment which may completely cure you, or in some cases he may not be able to do that for you. You often hear people say that, oh, I went to the doctor and he didn't have a clear idea of what was wrong with me. Well, it's often put down to the fact that some doctors are not as good as others. But the fact is that there are a number of situations in modern human health where it's often not clear what the disease itself is, and we don't have a full understanding of what the disease is. So he may not be able to make a very precise diagnosis of what is wrong. That's one issue. The second issue is that many times, even if he's able to make a very precise diagnosis, there are lots of illnesses in which the treatments that we have available is either not optimal, or in some cases there is no treatment at all. So what this then means is that we need ways and means of finding better ways to diagnose diseases, finding ways of diagnosing them faster, and also better ways of treating diseases. So, one of the things with children is that they fall ill very quickly, and they also recover very quickly from their illnesses. One thing that children often fall ill with is diarrhea. And there is a particular form of diarrhea caused by a bacterium called Shigella, which can become quite serious and in fact even affect the brain. And the most important challenge in treating a child with Shigella is to know that the child has that illness, because if you don't start the antibiotics as early as possible, you will not be able to offer an effective treatment before the disease or the toxins spread to the brain. The main problem here is that present methods of detecting Shigella take 48 hours. You have to culture the bacteria, characterize it and so on. And so as an example, if you had a way of taking a drop of stool or whatever and putting it into a tube, and if it gave you a yes or no answer as to whether the child has Shigella, then you could obviously help a lot more children than is possible right now. So I just use that to illustrate the fact that there are many challenges in medicine. Despite all the advances that we have, we can replace hearts, we can transplant them, we can replace lungs, but there are very, very basic issues in human health that still require understanding, and that understanding will likely give us solutions which can help people. Okay, so I noticed that the ages of the people in the audience is very, very broad. Some of you are obviously just out of school or in college. Others have seen a lot more of life. But the older people in the audience will remember a time whenever you talked about illness in India, the main thing you talked about was infectious diseases. You talked about malaria, you talked about tuberculosis. These are the main issues, and we talked about diarrhea. These are the main things that we needed to make our population healthy. But in the modern times, this has actually changed. Although we still have people who are ill with all of these diseases, the major challenges going forward are somewhat different. And there's a group of 3 diseases which already dominate and will continue to dominate in the years to come the health landscape of our country. And these 3 diseases are, in no order of importance, the following. The first is type 2 diabetes. There are about 400 million diabetics in the world, and unfortunately India is home to 70 million of those. The second is, of course, cancer. You always hear that we never used to hear about these many cases of cancer in the past. Now we hear about so many of it. Part of this is because it's easier to diagnose it now. There are better ways of picking it up. People are living longer, and that's the reason for it. And the third, very important group of diseases that is going to be a major health issue for our country is those diseases that relate to the brain, mental health disorders and other disorders relating to the nervous system of human beings. So today what I'm going to do is to focus on that last group of disorders, try to give you a perspective on those diseases. What is it that we mean when we say there are brain disorders and we need to find solutions for them? And also tell you 1 or 2 things that we are doing in the institute, more generally in the country, to try and find solutions for brain disorders. So Mike has already covered this, so I'm not going to go through this slide. But it's always good to see where the speaker comes from, and this is what NCBS looks like. If you do ever come to Bangalore, please come and see our very beautiful campus. It's as good as any facility you might find across the world. We also have a second and much newer institute on our campus, and that's the Institute for Stem Cell Science and Regenerative Medicine. It is just located adjacent to us, and the 2 institutes work together very closely on related problems. Okay, so this is a picture of a human being. You can see the outline, and everything that is marked in yellow is part of their nervous system. So you have on the top here, very readily recognizable, the brain, And right down the center of the person, at the back, is a spinal cord. And everything which is marked in yellow is a nerve. And all of these nerves pick up sensations from the surroundings and they transmit it to the central nervous system, which is the brain and the spinal cord. And that allows you to figure out what's happening around you, allows you to take evasive action to protect yourself from a hot object or a loud noise, and so on and so forth. So this is what we're talking about. Now, most animals have a nervous system, and the purpose of the nervous system is to have a very rapid way for you to detect what's going on around you and respond appropriately. And different animals have nervous systems of different complexity. And I just want everybody to just absorb just one single feature of this slide. These are all the brains of a number of different animals. The one here is a very simple worm that grows in the soil called C. elegans. It has only 300 neurons in its brain, and it's able to manage perfectly well with that. Here we have zebrafish, which has about 10 million neurons in its brain. And this is a fish, you know, that's a freshwater fish that grows with stripes on its body. This is a fruit fly Drosophila, which has about 10 to the power of 5 neurons. And I'm sure everybody has heard about the mouse, which has another order greater of neurons in its brain. So all these brains are fine for this particular organism, but the one that we are concerned about is the human brain, and it has about 100 billion neurons in it. This is the structure of the brain, this is the postmortem sample, and this is the cerebral cortex that allows you to have consciousness, to appreciate everything that's going on around you, and transmit signals to this part of the brain here called the midbrain. It contains the cerebellum, and that's what allows you to control your motor activities. So what we're trying to understand is this structure, how does it work, and when something goes wrong with it, how can we find solutions towards to those diseases. Please stop me at any point if I'm not being clear, because I'm not used to speaking in this sort of setting.
Please ask any questions you want at
Anytime,
any
if
point
I'm
in
not clear
time.
about anything, please stop me. So, what do we mean by diseases of the brain? Well, in some senses, it depends a lot on who you are and what your age is. But this is, every year the World Health Organization, through its subsidiary agencies, does a counting of diseases all over the world. They count every type of disease in every type of country that there is, and they publish a catalogue. And this is the result for India for 2016. And this relates specifically to the nervous system. So the vast majority of problems in the brain are in fact strokes. I'm sure you've heard about people who have strokes. The blood supply to the brain is affected, and that part of the brain stops working, and because the brain cannot regenerate itself, you're left with a deficit. So sometimes you hear of people who cannot move an arm or a leg and so on. And that's because a part of their brain has stopped working. The next major group is dementias. So this is the issue of people starting to forget things. Not our regular run-of-the-mill forgetting to buy milk or whatever, but much more serious things, forgetting their relatives, forgetting the way back to their house, and so on and so forth. As you all know, the life expectancy in India is much more now than it is before, and it's on the rise all the time. And as a result of which, the incidence of Alzheimer's disease and other types of dementia is now increasing. So this is going to be a major health problem in the next 50 years as more of us live longer and get to older ages. And then there are another group of multiple other disorders, each important to the person who suffers from it, but in terms of numbers they are very small. So things like Parkinson's disease, you may have heard of it, it's a movement disorder. You have heard of motor neuron disease, its most famous sufferer, Stephen Hawking, who couldn't move and then ultimately died some time ago. And epilepsy, which is very common in children. And then various infections of the brain, viral infections, bacterial infections that affect young children. And depending on your age, you are more likely, if you're a young person, to suffer from an infection of the brain. And as you get older and older, things like strokes, dementias, and so on take over. There's one group of disorders that I've not yet had a chance to mention, and that's something that is very, very common in the population. So all of the disorders that I mentioned just before this are associated with specific parts of the brain. But these disorders mentioned over here, who go by the general term mental illness, are not linked to any particular part of the brain. They affect the whole brain. And I'm sure that you've heard of all of them. Depression, people have heard of somebody or the other having depression. You've heard of anxiety disorders, people are very anxious, they're unable to focus on their work because they're just thinking about, worrying about things. Everyone's heard of substance abuse or substance dependence, which in India largely translates to alcohol dependence. And then there is something called bipolar disorder, where the person swings from being very depressed on one day, and a few days later he may swing to mania, which is a state of being extremely aware and hyperactive. And then there is schizophrenia or split brain disorder. And then there's a very minor amount of eating disorder. But you can see the numbers, they're quite impressive. 3.5% of the population of the country suffers from some form of clinical depression. And if you consider that we have 1.3 billion people in India, that's quite a lot of people to deal with. But let's just look at the absolute numbers to understand.
Sorry, I just wanted to know, how's eating disorder going to any of these? So it's like all rest of them fall in one
Okay. Yeah. So this thing is a census of illness. So it's just like the census is done in the general population, the WHO asks people to go around, doctors, patients, whatever it is, and just gives them some criteria and say, you have this, you have that, and so on. That's how you come up with these numbers. A person can have more than one of them, and it doesn't exclude somebody with depression may also have an eating disorder, and the other way around. So this is just for the sake of conceptualizing what the problem is. It helps governments to plan health priorities, what should be done and how. So it's not really so watertight between all these compartments. Okay. So BPAD, for example, this number looks really very small, 0.5%. You might wonder, why should we worry about 0.5%? But if you have 1.36 billion people, that's 6.5 million people who suffer from bipolar disorder. And these are disorders that affect young people, people who are in the prime of their life, who are taking decisions about their careers and so on. And very importantly, all of those diseases have a prolonged course of, prolonged life history. So they run for 15 years or 20 years and so on. Even in the city of Bangalore, there must be about 60,000 people with BPAD. So, you get an idea of the scale of the problem, and I really just want to emphasize why it is that we need to find solutions to these types of diseases.
In this slide, the slide previous to this, percent somewhere.
Yeah.
Not this.
The one before this.
Yeah, there is not here. 67%. So, how this 0.5% related to the 67%, where you want to concentrate more on those 60,000 people concerned, I don't get with this.
So, to a person suffering from an illness, what you suffer from is most important. It doesn't matter that there are 67% of the population will suffer from a stroke or not. If you have BPAD or if you have an eating disorder, that is the primary dominant thing in your life. So I don't want to say one is more important than the other.
I'm just thinking that
as
the government is concentrating on a leaf and forgetting about the whole forest.
May not be true. The government, of course, so one of the reasons why these kind of statistics are produced is that every government, whether it's Indian or otherwise, needs to decide how to spend its money, what should be the priorities and so on. And up till now, the focus has very largely been on infectious diseases for the reasons we discussed at the beginning. But these sorts of numbers tell you that going forward, we're going to be having a very big problem with brain disorders in general, including strokes and many other things. In part, this is because life expectancy has increased a lot, and there are many more older people around now. than there were before. And I'm sure that everyone's noticed that there are many more older people around than, let's say, when 25 or 30 years ago. So it's not that strokes are more important or less important. It's just
Depression across the world is the worst disease now, mentally speaking.
should be.
Go ahead.
I mean, I think if you ask different people in the room what is the most important disease, I think there might be maybe slightly different answers. But you know, it is very difficult to ignore the fact that 67% of all brain disorders in India are a stroke, that somebody who's middle-aged to old is not. So I just want to just make the audience aware that, you know, we have two groups of diseases here. The ones on the right, the ones on the left which are marked in red, are the so-called brain diseases or mental disorders of the young. They affect young adults. And it's a problem that affects them right from the time they're about adolescents onwards and young adults. And usually, if you're going to develop this, you would have developed it by the time you're about 30 years or so. And on the right-hand side, marked in blue, which you can't see, are the so-called brain disorders of the elderly or the older people. Alzheimer's disease, this is dementia, Parkinson's disease, which is a degenerative movement disorder, and Huntington's disease, which is also a degenerative movement disorder, but of a different type. So we need answers to solve, we need answers on how to deal with these conditions, find better ways of treating them, and if we can't treat them, at least better ways of managing them. Sometimes, you know, if you can diagnose a disease earlier, even if you can't cure it, you can manage it better, and perhaps it won't progress as fast as it could otherwise do. So, the history of people trying to figure out what is wrong in the brain is very old. All of you have heard of Leonardo da Vinci, the famous polymath painter, artist, and so on. One of his great fascinations was the human body, and he was particularly interested in the brain, and he developed many methods to give us. And he discovered that there is a part of the brain that specifically controls speech. It's now known by the name Broca's area, and it's in the left frontal lobe. And then Jackson and Carl Wernicke, by studying patients with epilepsy, worked out which part of the brain controls the movement or which specific part of the body. So the point I'm trying to make with this slide is that research helps you to understand how the brain is organized, and that also then helps you to work out how you might treat a disease affecting that particular part of the brain. Okay, so the final output of the brain is behavior. The way I'm interacting with you and the way you're interacting with me is the final output of how the brain is functioning. But this final output of the brain is organized at multiple levels. Just as there is a structure to many things in life, this is the structure of the brain. The behavior that you see is because of the activity of all the neurons in my brain, and all the neurons in my brain are connected together in specific ways. If there is a miswiring or miswiring, I should say, then you will find my behavior unusual, beyond the norms of society. And those circuits are put together by individual cells, and if there's anything wrong with one of those individual cells, you still get a disease of the brain. Now, the first three levels of this, which is from behavior up to circuits, you can get information about it without having to touch the brain. So that's what I want to convey in this slide. So you can get behavior by going to a neurologist or a psychiatrist. He will examine you, he will make certain observations, and he'll work out whether your behavior is beyond the norms of what is expected in present society. Then we have very powerful tools that allow you to look at the structure of the brain. I'm sure everybody's heard of CT scans and MRIs. They're extraordinarily sensitive these days, and they can allow you to look at the structure of the brain from outside and work out if there are any structural abnormalities, or sometimes even, why did you come and sit here?
There's a seat in the front.
So, many times, we talked about strokes a few minutes ago. One of the first things that happens when somebody has a stroke is that the neurologist, although he can more or less say where it is, would like to know how big it is, how reversible it is likely to be, and so on. And what he does is ask for a CT scan or an MRI that tells him that this particular part of the brain has been deprived of blood and is now affected. Then there are scanners that allow you to even monitor the activity going on inside the brain. So these use things called radioisotopes, and they allow you to see which parts of the brain are active, is the activity abnormal or not. And this, for example, this what is shown over here is a normal behaving brain being imaged from outside. You are touching it. The yellow regions are the regions of high activity, and this is a similar scan from a brain of a person who is depressed. So, you can get broad information about how the brain functions by these types of methods. And then, this is clinical electrophysiology, electroencephalogram, where you put electrodes on the outside of the brain. And this is used to work out why somebody is having an epileptic seizure and so on and so forth. What you will, however, see is that all of these things only allow you to indirectly infer what is going on in the brain. But perhaps the most unusual thing about the brain is the following. Now imagine that you have anemia, or you have some problem with your liver or your kidney. You go to the doctor, he does the usual blood tests, and he decides, I'd like to have a look at what the kidney looks like, or I'd like to have a look at what the liver looks like. And so what does he do? He uses these sophisticated methods to take out a small part of the liver or the kidney, and then it's examined under a microscope. Somebody in the audience is a histopathologist. Such people look at it and they make diagnoses about what is wrong with that particular tissue. But reflect on this point, that in the case of the brain, there is no part of the brain that is dispensable. And I cannot offer to anybody a piece of my brain as a biopsy for diagnosis. And what that means is that until very recently, there was no way for us to look at the cells of the brain and to work out what is wrong with them until the person had died. You can only do it post-mortem. And this had been a major challenge for many, many years. And what I'm going to tell you about in the next 15 minutes or so is some new technology that we are using, which has become available in the last, let's say, 5 to 8 years, that allows you to now actually do this. Have the cells of the brain in the dish in front of you and make observations on what's going on in them, what's wrong with them.
You were talking about active, brain is active. What exactly?
So usually in these kind of scans, what is done is you go to the diagnostic center or the hospital. They put your head inside one of these scanners, which has a magnetic field in it, and then they ask you to do some task. They might ask you to raise your hand or whatever it is, and then when you're doing that, there's some parts of your brain that neurons are actually active, and when they are active, they emit a signal which is captured as yellow over here. And this is done using a form of radioactive glucose, because when a neuron is more active, it uses more glucose, and you can get a signal out of it. So that's what the scan does. So the technology that allows us to do this involves stem cells, which is the advertised theme of today's talk. During the development of a human being, we go from being a fertilized egg through a series of steps where the single cell divides to give you more and more cells. And not only does it give you more and more cells, but the cells become differentiated, shall we say, and there are now different types of cells present. So up to this stage, which is a 16-cell stage, every cell is the same as every other cell. But thereafter, you start seeing different types of cells. So you start seeing some muscle cells, you start seeing skin cells, you start seeing brain cells, whatever, intestinal cells and so on. And in fact, even by about 10 weeks, all the cells are there, and thereafter all the development that happens during pregnancy is growth in size and some motor differentiation. But people have tried to understand for a very long time how this works, and this is the field called developmental biology. And people are asking the question that although all these cells have the same DNA, what allows them at this point to start behaving differently, to become a muscle cell or a skin cell or an intestinal cell? So this is something that's been studied for 100 years, and what we now know is that the single cell that we started out with after fertilization ultimately gives rise to about only be one type. But this is a field in itself and is worth a science cafe in its own right. But what I want to tell you is that until about 15 years ago, we believed that this process was completely irreversible. That is, you couldn't go backwards in this cascade. So you can start with a fertilized egg and you can make all these cell types. But you cannot start with any of these cell types and make the fertilized egg. Well, that changed about 12 years ago when Dr. Shinya Yamanaka, who's shown here on the left, found a way of taking cells from any part of your body, so skin cells or white blood cells from the circulation, and he was able to treat them in a particular manner so that they now return to this state. Okay? So in fact reverse the ability of the differentiated cell and made it become a so-called pluripotent cell. I don't want to go into details of how this is done. If there's anybody who really wants to know the details, I'll certainly point them to a reference. But imagine the implications. You could now take from any one of us a piece of skin, or I could take from you a sample of blood and isolate white blood cells. And using Dr. Yamanaka's method, I could convert it back into a stem cell, which is what this top cell is called. Okay? And once you have the top cell, you can now, using a knowledge of developmental biology, convert it into any tissue you want. And this has now been done. People have made various organs out of it. People have made an eye. People have made skin. You can make cardiomyocytes, the cells that allow your heart to contract. There's even somebody who's making so-called vegetarian meat using this method. So they're growing muscle cells in a dish so that you can have that, and so on and so forth. But for the purpose of today's talk, what this means is that you can now do the following. Imagine that there is a patient who has some brain disorder, and you would like to know what is wrong with the individual cells in his brain. How could we produce this in a dish? Because to do experiments and to make observations on individual cells, you need them in the dish. Yeah? So what is done these days is to take somatic cells, as they are called, skin cells are usually what is used, or blood cells, and then you do something called reprogramming, which is what Dr. Yamanaka discovered, which is supply them with certain goodies, and what they do is that they de-differentiate into something called induced pluripotent stem cells. And I'm sure that you've heard about this term in the popular press. And then starting with these induced pluripotent stem cells, you can then differentiate them and to make whatever tissue you want. Once you have that tissue, there are many things you can do. You can do something called disease modeling, which is you observe the cells of the brain and try and work out what's wrong with them. The other thing you can do is you can do drug screens. So, in the old days when people wanted to discover a drug, they would collect 50,000 rabbits or rats or whatever, try out all these compounds on them. It has various issues with regard to animal ethics and animal rights and so on. But now you can do this in the dish using specific tissues of your interest. So that's one thing you can do on those cells. The two most sensitive tissues in the human body for most forms of toxicity are either the liver or the heart. And so if you produce cardiac cells or liver cells in the dish, you can now apply the drug and ask whether it has any detrimental effects. It saves money, it saves time, and in the United States, the Federal Drug Administration, which determines what can be sold as a medicine or not, now requires that all new drugs are tested using this approach and not just using animal testing. So this is what is allowed. And so you can then imagine the possibilities. I think that there are many people here who must be thinking, what can I do with this? Actually, there are many, many things you can do. I will just try and show you some images that sort of tell you that this is not all just fairy tale, but actually happening in the lab. It's happening in Bangalore and in many other parts of the world on a daily basis. These are induced pluripotent stem cells. They are growing in a dish in my lab, and they have been stained with various markers to prove that they are actually pluripotent stem cells. So they're growing nicely there in colonies like this, and this is some experiment to prove that they can give rise to any of the tissues in the human body. By the way, these are human cells, and they're karyotypically normal as well. That is, they have no chromosomal abnormality. You can take these iPSCs, and then using our knowledge of developmental biology, you can produce neurons. Who has heard of any other type of cell in the brain other than neurons? Yeah. So, for as long as we've studied the brain, everybody thought that the neuron was the most important thing. And there are, 10 to the power of 11 neurons in the human brain. But in fact, there are many more cells called glial cells, which until about 5 years ago were thought to be supporting cells. That is, these are cells that provide nourishment for the neurons, which do the real business. But these days, if you look at any of the major journals, every month you see a new paper which says that neuronal function depends critically on glial cell function and so on. And many diseases, the primary defect is now being shown to be in the glial cells, and the neurons are being affected in a secondary manner. Anyway, there are methods to produce all the different types of glial cells and neurons in a dish in your lab, and you can now study whatever it is you want to study with them. You can also
small this differentiation of neurons, differentiation of iPSC into neurons, astrocytes, oligonucleotides. What is the direct to get transformed into briefly can you just
yeah, so before
what factors are important to make the specific of results?
So before iPSCs came along, people were more generally interested in the problem of how does the brain develop. And this is the field of developmental neurobiology. And using models such as the mice and so on, they had worked out that there are certain growth factors and there are certain differentiation factors that are required at specific stages of development to give rise to particular cell types. So when you start with iPSCs, the first thing you make is not a neuron, but something called a neural progenitor cell. And that neural progenitor cell can subsequently differentiate to give you some of these cell types. And we now know molecules that can drive a cell to become a neuron from an NPC. We know specific factors that can drive it to become an astrocyte or an oligodendrocyte and so on. So when we're doing experiments in the lab, we are borrowing from that knowledge and adding these factors into the culture so that differentiation happens in indirected ways. The last and most remarkable thing is that one can now produce something called brain organoids. I'm sorry I don't have a picture to show you of a brain organoid, but basically it looks like a tiny brain. You can differentiate it in the dish starting with an iPSC, and these days it is possible to maintain them in culture for about 8 or 9 months, starting from the day of the culture. And an 8 to 9 month old brain organoid is approximately equivalent to the brain of a newborn child. So we now have the ability to model the development of the brain starting from fertilization all the way up to roughly about the end of gestation and birth. So this is what they look like in the dish.
So when you say that when you are able to generate this, is it only parts you are able to generate or the some two things together? Are we able to also
Yeah.
simulate?
So the brain has many regions. First of all, it's divided into forebrain, midbrain, and hindbrain. Forebrain is only a feature of primates and humans. Others are. Yeah. So we know the transcription factors that are required for each of these regions to develop. And by providing them in our culture conditions, you can make the brain differentiate into particular parts. So some people might be interested in studying Parkinson's disease. It's a topic of great interest. Parkinson's disease affects a nucleus of the brain called the substantia nigra, which is part of the midbrain. So when you're trying to produce these cultures to study Parkinson's disease, we would do so that we direct the cells to go towards midbrain type of structure. When you want to study something which relates to the cortex, the folded thing that you see in the picture, then you would do something called forebrain
Does this
differentiation.
include even the brain barrier?
No.
No.
So one issue with brain organoids right now is that it doesn't include the blood-brain barrier and it doesn't include any blood vessels. So the extent to which you can grow it is in fact limited by how much blood supply, because as it gets bigger and bigger it needs nutrients, and normally nutrients are supplied by the blood vessels. So we still don't know how to make the vasculature grow within the developing brain. The reason for that is that the vasculature comes from endoderm and mesoderm, and the brain comes from ectoderm. So we haven't yet worked out how to do that. When I say we, it doesn't mean me, but a huge number of scientists across the world who are working on this. So this is how it looks like under the microscope. As you go more and more days into development, you see markers that are usually seen in the brain. This is beta-T-tubulin. It's a part of the skeletal structure of the neurons. This is neurofilament, which is another protein that is very specific to neurons. It appears by day 60 and so on. And if I can make this movie work, you will be amazed at what you see. So what we have here are neurons that have been in culture for 90 days. So that's 3 months. We can do it up to 120 days as well. This is a living culture. And it's been loaded up with a dye that allows you to detect the activity of the neurons. And I'll try and play the movie, and hopefully what you will see is you can see flashes of light going on and off in different parts of the culture. And that represents the neurons firing and their activity picking up. So I'll try to do this now. I hope you can see different parts of this image. There's one, there's another one, there's one here, another one there. So you can see the lights going on and off, and that as it becomes brighter, the neuron is being activated, and once the color goes down, the neuron's activity is going down. So these are 30,000 or so neurons. They're all connected to each other.
What is the engaging technique usually?
So this is a fluorescent indicator. It is measuring ion called calcium, which is present inside the neuron. And from everything we know, calcium is one of the central messengers of activity in the neuron. So we are actually monitoring the calcium signals.
Even for only that hardware and not the others.
So here I'm not providing any stimulus. This is spontaneous activity. So if you think about it, during development in utero, I'm not sure what the stimulus is, but neurons progressively get activated as the brain develops. They must be expressing proteins that make them active, and that's dependent on environmental factors as well as developmental issues. So this is only 120 days, it's not very much, maybe about first trimester or something like this.
So, I'm still thinking about the question we had asked about developing the brain. You said vasculature is a promise. So, if we know how to make the organoids, and if we know how the brain develops from the fetal stage to policies with both organized and the national?
It should be,
Has that
not been done.
been attended?
Not been done.
If that's done, then would you
Well,
think
ultimately,
it's possible to?
ultimately the problem with present technology is that, the brain is the most metabolically active organ in the body, energetically active. And if you want to keep it alive for beyond 7 months, with all of this activity going on, 10 to the power of 11 cells, I think the most challenging thing is going to be two things. One, providing enough nutrients so that it doesn't run out of nutrients. And second, providing something called cerebrospinal fluid, which is what normally removes the toxic substances from the brain away. So we don't have cerebrospinal fluid and we don't have vasculature yet. These are the things to aim for in the future. For example, somebody was asking about stroke. If you want to study stroke, obviously you need blood vessels in there. That is something to look forward to in the future. Okay. This is probably my last or second last slide. Now, unlike blood cells and so on, one of the key features of the brain is that all the neurons are connected to each other. Okay? And forming these connections and breaking these connections is the bread and butter of any brain, of any nervous system. Whether it's a worm or a mouse or a tumor. And many people, the human brain is very unique in that it is one of the few brains in which development continues after birth. So the development of the cortex in the human brain, the formation of the connections, that continues all the way up to adolescence. And so anything that influences the environment that influences that will have an impact on the development of the brain, will have an impact on the development of the child, and so on. So what is the surrogate that we can use to monitor that? And this is what has been done over here. What has been done is to take neural stem cells and put them out in a tissue culture dish. And what happens when you trigger differentiation is that they start putting out these long processes which you can see. Can everybody see the processes? The round thing is the cell, and the thing sticking out of here, these processes are so-called neurites. And the neurites are what gives rise to the long axons that connect one cell to the other, one neuron to the other. And what is marked in green are all the individual neurites. And as time goes on, 6, 12, 24 hours, the neurites get longer and longer. So you can now take such cultures and measure how fast the neurites are growing. You can measure how fast they're branching, how well they're making connections to each other, and that allows you now to look at whether these defects exist in particular genetic mutations or in people with particular disease conditions. What's shown over here at the bottom is a neural stem cell from something called DISC1. DISC1 stands for disrupted in schizophrenia. It is a gene which, when mutated, gives rise or is very highly correlated with the development of schizophrenia in humans. And what you will see is that actually here the neurite outgrowth is much less than in the normal one which is shown over here. So I just wanted to give you a flavor of the types of things you can do with this technology to try and understand what's actually going on in the individual cells of the brain that may be manifest in the electrophysiology, that may be manifest in the MRI, and that ultimately is manifest in abnormal behavior that we describe as mental illness. So this is a big problem, and mental illness is really a major issue in the country. And some time ago, the government of India decided, actually, the Prime Minister Narendra Modi went to Japan, and he was introduced to Dr. Shinya Yamanaka, and he decided that, well, this is a new technology, and why don't we try and use it to solve some problems in India. So he came back and asked several people, what can we do? And one thing that was put forward was that, why don't we use stem cell technology to try and understand human diseases. And that is the origin of this BLiSc program which is mentioned over here. It's funded by the Department of Biotechnology,
the Government of India, and it's a collaboration between INSTEM, NCBS, and the National Institute of Mental Health and Neurosciences. And these three institutions and their scientists are coming together to try and use modern methods to understand what might be the underlying basis of mental illnesses. So if you want to find out more about this program, please do visit our website, which is over here, and you can also write to this email address if you have any questions.
I have only one question. We discussed about adolescence and the neuron development. We see a lot of depression and other things associated with either a drug use or some other habits. Okay. Is there any insight into that?
Stem cells by themselves cannot obviously solve this problem. Stem cells are only one component. The use of stem cells is only one component of the work that needs to be done. The ADPS program actually has three main verticals. One vertical is very deep clinical characterization of these patients who have many of these illnesses, including substance dependence. And what can we look forward to from that part of the program? Well, what that part of the program does is, first of all, I should say that for the first time in India, somebody, not just scientists but funders, have had the foresight to devise a program that runs for 20 years. Okay, so it's a 20-year program. It's called a prospective study. That means you start today and you go forward 20 years into the future. Patients are studied every 3 years. So it means that you can watch the disease developing. Those who don't have it will develop it. Those who have it may get better or may get worse. And just this clinical information, if it is collected appropriately for the full duration of time, will give clinicians a lot more tools to answer the types of questions that you're asking. You know, Alzheimer's disease, I think somebody mentioned it earlier. A big challenge in Alzheimer's disease is actually knowing it quite early. It's not very helpful to know that somebody has Alzheimer's disease when the symptoms are well advanced. Even the lay person can say that they cannot remember whatever. So some studies of this type that I just described done elsewhere in the world, and we now know that there is a part of the brain called the hippocampus, and changes in the MRI in the hippocampus can be seen at least 15 to 20 years before a person manifests with memory loss. So if you know that much earlier, you can at least plan on how you're going to treat such individuals. In some cases, there are genetic basis for it as well. So I think that, you know, when you talk about using these technologies to deal with the problem of brain disorders, one must not always think that it is about a cure. Sometimes it is about making discoveries that allow you to manage a patient better. Manage patient better for his or her own outcome. Manage a patient better for the caregivers who have to look after the patient. So that's what I think is another thing that you should look forward to from this type of study. It's not always about curing illness. In any case, in clinical medicine, what you are supposed to aim for is the best quality of life for the patient, and that's what I think you can get at using these studies.
Well, is helpful in algorithm diseases identified or embryo anomaly in these modern techniques?
I didn't fully understand it. Could you just repeat it again?
Will it be helpful in diagnosing a congenital disease in an embryo or anomaly scanner?
So we already have many powerful technologies that allow us to diagnose abnormalities in early embryogenesis. You may have heard of something called scope and diagnose cytogenetic abnormalities. I don't think these technologies are required for it. So the way that neurons function is by firing something called action potentials, and that involves pushing sodium ions into the cell and potassium ions outside. So this is not by diffusion but against diffusion. So there is a pump that uses energy that is present in the plasma membrane of all cells, but neurons probably use it 100 times more than a lymphocyte or a skin cell. So that's where the requirement for energy comes from.
Three situations. One, normal. Second, sleeping. Third one, meditation. Will there be any significant changes in the brain function?
First of all, I'm not competent to answer this question, but I do know what you're talking about. It refers to a field called neuromodulation. So neuromodulation refers to, excuse me. So neuromodulation refers to all those things that you can do from the outside that may change the activity of the brain beneficially. The oldest version of this, which is quite unpopular, is something called electroconvulsive therapy, where you give a shock to the brain, it resets the neurons and the person may or may not recover. But there are more modern forms of neuromodulation. Meditation is considered to be one of them. Yoga is considered to be another one. But what is lacking is systematic studies that are able to explain why these things are beneficial. I think that we have all heard that yoga is beneficial. We have all heard that meditation is beneficial. They must be acting in the brain through changing the activity of various parts of the brain. And there have been no systematic studies to address these. So at NIMHANS there is actually a specific center for neuromodulation that is carrying out systematic studies of the impact of yoga and meditation on the brain to try and take it from the lay public's imagination into a more formalized scientific explanation of why these things are. So we await the results of that.
So, is it possible to change the connections of the circuits so that the required behavior could be either by medicines or through innovative stimulus?
Not in humans. This experiment has been, it's a very interesting question that you asked.
Especially for children.
Can you just repeat it so everybody can hear?
She's asking if it is possible to rewire the brain to get desired behavior. Is that what you're saying? So this experiment has actually been done many years ago in ferrets, which are a type of mammal. Rodent. So what was done is to take the nerves that normally go to the, every sensory organ in our body projects to a particular part of the brain. So your eyes, the nerves from that go to the occipital lobe, which is right here. The ones from your ear go to the parietal lobe where there is an auditory area. So there was a scientist Dr. Raghu, who back in the early 90s did an experiment where in young ferrets he connected the nerves that normally go to the eye, from the eye to the brain, to the place where the auditory connections go. And now the auditory cortex starts behaving as though it is the visual cortex. So this is an extreme example of what you're asking. I mean, I don't think that this is possible in an adult. First of all, this was possible only because it was done in a very young animal where connections were still very plastic. one of the most unfortunate things about the central nervous system is that nothing regenerates in it.
For like children with dyslexia, like some connections could be changed in
Yeah.
their behavior.
Yeah, so this falls into the same category of question that he was asking. For example, when you have an autistic child, it is often recommended that certain types of environment or certain types of behavior or certain types of exercises are beneficial to a long-term outcome. It is assumed that when you do it in a young child, this somehow positively influences the connections in the brain. As I said, the human brain is the only one we know of where so much of development takes place outside the uterus. So after birth, up to the age of 15, the brain is still developing in a human, connections are being made and broken and so on at a very high rate. So during this sensitive period of time, I think if you had interventions, they might work, but again, it goes into the same category of answers that I gave him, which is that we know that it works, but we don't know how it works. So I don't want to say that there is rewiring, but there probably is.